This video is going to focus on the human tissues in terms of histology. Now, as a reminder, you should be utilizing your module reading guides and exam review guide to help streamline your studying for your upcoming exam. So our focus is going to be the study of tissues, and there are four primary classes of tissues that we talk about when it comes to humans and other animals. Now, you need to make sure you understand the definition of a tissue, which is a group of similar cells and their cell products that work together to perform a common function, typically in the form of an organ. Now, the four primary types of tissues we're going to focus on are epithelial, connective, nervous, and muscular. And the overarching differences between them have to do with the types and functions of their cells, as well as the characteristics of the matrix or the extracellular material, and the relative amount of space that are occupied by the cells versus their matrix. So before we get into each of the different tissue classes themselves, I want to take a moment to address what I mean when I say extracellular material. So the extracellular material is all of the material that exists on the outside of the plasma membrane of a cell. And if you remember from our last module, that membrane is what separates the internal workings of the cell from the external features. Now, the extracellular matrix includes fibrous proteins such as collagen, that's pictured here, and it also includes this clear gel, which we often refer to just as fluid, but it's also referred to as the ground substance. That fluid is also sometimes referred to as the extracellular fluid because it's the cellular fluid outside of the cell. The other term that we'll use for it is interstitial fluid, which literally means the fluid between the tissues. So that is the term we tend to use more when we're dealing with transport. But remember, extracellular matrix includes proteins that are these fibrous material, and then the fluid component to it. The other thing that I want to address before we get into the specific tissue types is where do these tissues originate from? And we refer to them as embryonic tissue before they start to differentiate into their main functions. Now, the picture, you'll see that there is a type of tissue identified in blue. The tissue that's identified in blue is the ectoderm, and the ectoderm is the tissue that ends up being differentiated into the outer layer of our body as well as our nervous system. So you're thinking of epidermis of your skin. It can be the sweat glands, the hair follicles. It can also be the nervous system, including your nerves themselves and your brain and spinal cord. The red tissue that you see doesn't actually form immediately. It's once the blastula, which is the early embryonic stages of development, starts to get invaginated, and you'll see these red cells start to migrate more towards the middle of everything. And it's literally the middle layer of the embryo. These cells will end up differentiating into your skeletal muscular system and our circulation and lymphatic systems, excretory and reproductive structures, and the dermal layer and below of our skin. And the yellow tissue that you're seeing developing is actually creating this tube from the mouth to the anus, which is your digestive tube. So it's basically any part of your digestive system. It's normally the epithelial lining associated with organs like your liver and your pancreas, your respiratory system, excretory, urogenital, and some of our other endocrine glands, such as the thymus, the thyroid, the parathyroid, and that kind of thing. So the reason why we bring this up is because as we start to look at different systems through the progression of AMP, you can relate it back to the original development and the fact that these embryonic cells are the things that differentiate based on gene expression and location to give rise to all these different tissue types we have. The other thing that I want to bring your attention to is when we look at histological images, that we are looking at a three-dimensional object two-dimensionally. So the first picture, I think, does a really good job showing that if you've got a whole cell, and we're looking at an egg for this one, that depending on where the sagittal cross-section is taking place, you may see just the egg white versus the yolk itself. And as you progress through this, in order to get both the egg white and the yolk, you have to be more medial than lateral. Now, image B is showing what happens if you have something that is a slightly different shape. This one's demonstrating what happens when you look at a tube, depending on where the cross-section takes place. And then image C is also showing a tube that if you were just to take a longitudinal cross-section of this, you would get a lot of different variations with it. So when we're looking at the histology images, you have to remember that we're looking at a very thin sample of the entire three-dimensional structure. The other terminology that we often refer to when we're dealing with these different histological images are how the sections are actually cut. A longitudinal section will be a tissue cut along the long axis. A cross section or a transverse section is when the tissue gets cut perpendicular to its long axis. And then an oblique section is when you get a cut at an angle between the cross and the longitudinal sections. So just depending on what we're looking at, you may be observing different types of sectioning. So let's switch gears, and instead of doing the general overview, we're going to jump into epithelial tissue. So the image that I have for you is actually showing different samples of epithelial tissue throughout a mammalian system. And you'll notice right off the bat that all of them have slightly different shapes and slightly different stratification to it. And I'll come back to the terminology that's associated with it after I describe this in an analogy format. So I like to describe epithelial tissue using, I call it the hose analogy, where if you were to actually take a transverse section of a hose and you're looking at it end on, that the water is going to move through the central portion and your hand would come in contact with the external portion. In histology, we refer to an open space, which is typically fluid filled inside of a cell or inside of an organ, versus the external area, which would also be surrounded by some type of fluid, as lumen. And epithelial tissue is tissue that is lining lumen. And normally, because it is lining the internal and external components of an organ, it has a barrier function to it. And the reason why it works as a barrier is because the cells are so tightly packed together that material has to move through the layer of cells in order to get to any material underneath it. So the other terminology that we reference when we're dealing with epithelial tissue is the fact that epithelial tissue, because it is a lining, has one side of the tissue exposed to the internal or external environment, and that's referred to as the apex. So anywhere lining the direct lumen would be the apex, and all the cells that are tightly packed together are connected to what we refer to as a basement membrane. And that basement membrane in all of these other pictures is indicated by the blue on the sides of it. And that blue basement membrane is the anchor point. And we also refer to that as the basal surface. But of the 6 different imaged here, you can see that there is a different type of tissue underneath the blue basement membrane, and that's your connective tissue. So that basement membrane's function is literally to adhere your epithelial tissue to the connective tissue underneath it for an anchoring point. Now, the other neat feature about epithelial tissue is that it's avascular, so it doesn't have blood flow to it. In order to get nutrients to this tissue and get waste materials away from it, we rely on the vascular nature of connective tissue that is just below that basement membrane. So when we're looking at the actual examples over here, you can see the apex on all of them is going to be the top area, with the exception of your simple cuboidal. The apex is the inside of the tube. The basement membrane is that blue membrane that you see down at the bottom. With the simple cuboidal cells, you'll see that it's actually the external portion of it. There would be other connected tissue on the outside of this. Now, the other thing that you'll notice is that there's a shape difference between these. There's things that are listed as cuboidal, which cuboidal cells, the height from the basement membrane to the apex is about the same as the width of that cell. So it resembles a cube shape. The nuclei of cuboidal cells also looks very circular typically. This is in contrast to squamous cells, where squamous actually translates into scales, but they kind of look like squished or fried eggs to me, where from the basement to the apex, it's flattened and it's wider than it is tall, where the columnar cells resemble columns because from the basement to the apex, they're taller than they are wide. Now, the other variation that you'll see is that we have some that are listed as stratified versus simple. Simple refers to having one layer of that cell type, and epithelial tissue is literally named based on the number of layers you see and the shape. So simple squamous would be one layer of squished cells. Simple cuboidal would be one layer of the cube-shaped cells, and then simple columnar would be one layer of the column-shaped cells. As far as functions go, if it is simple, meaning there is one layer to it, it typically is an absorptive or secretive function, where we're trying to let material pass through this more easily. Simple squamous, you actually find the lining your alveoli of your lungs, as well as our capillaries, which allows for easy diffusion of oxygen and carbon dioxide. The simple cuboidal cells are found in areas like your kidney, and they function more in filtration, where it secretes some material, but it also reabsorbs other material as we're forming urine from filtering our blood. And then the simple columnar cells typically is found within the intestines, where these would be absorptive functions. Now, the simple columnar cells, you'll actually see better images in your textbook that show that they've got microvilli to them that help increase the surface area. So they definitely have more of an absorptive function. Now, when you have more than one layer of cells, we call it stratified. Stratified cells typically are more protective in nature. They will allow for some absorption and some secretion, but the primary role of those is going to be protective. Stratified squamous you find within your throat, your cheeks, and those are areas that are constantly having some type of abrasion to them that help to protect those areas, and they're constantly going through mitosis, replacing those cells from the basement membrane up. The stratified columnar cells, you actually find these within the urethra, which there's a lot of movement of material through that area as well. So again, a protective function. Now, the one unique one that you'll see up here is the pseudostratified ciliated columnar cells that we find in our nasal passageway and along our respiratory tract. These are cells that they look stratified, but it's a false type of stratification, hence the pseudo-stratified. Even though you can see that the nuclei are staggered, if you look from the apex to the basement membrane, every single cell connects to that basement membrane. So the nuclei being scattered is what makes you think they're stratified when they're truly not. The ciliation of these particular cells also help to move mucus and debris out of our respiratory system. So that way we don't actually get it down into our lungs. So the image that your textbook actually uses is the one where it's showing the classes of epithelium as whether simple, pseudostratified, or stratified. So it's literally based on the layers that you're seeing. Simple meaning one, stratified meaning many, and then pseudostratified is the intermediate where they're truly simple, but they look like the nuclei are at different levels. And then they're also named based on their shape. So your squamous cells are the squished ones. They're shorter than they are wide. Cuboidal are your cube-shaped ones where they're almost equivalent height and width. And then the columnar are going to be taller from the basement to the apex versus their width. Now, keep in mind that the functions of your epithelial tissues are going to be based on their overall class and shape. The primary functions of epithelial tissue in general are to protect the deeper tissues from injury and infection. They also produce and release chemical secretions. They can excrete waste. They can absorb chemicals, including nutrients. They can selectively filter what gets into or out of a tissue. And they can also sense stimuli. So there's lots of important functions of your epithelial cells, but remember, they are lining all of the other organ systems internally and externally. All right, the next group of tissues that we're going to focus on are the connective tissues. And I like to use this image for the overview because connective tissue is a larger class of tissues that what they all have in common is that the cells occupy less space than the matrix. So they have a lot of extracellular material and the cells are spread farther out. So as far as the role of connective tissues go, it's a supportive, connective, and protective function. And it also depends on which one you look at. But connective tissue, in contrast to epithelial tissue, is almost always vascularized, meaning that there's a lot of blood vessels that run through those connective tissues. The only exception to this particular rule has to do with your tendons and your ligaments and cartilage. So tendons and ligaments are made up of it's made up of fibrous connective tissue that doesn't have a lot of blood flow to it. And cartilage also has few or no blood vessels. The reason I'm pointing this out is because if you were to sustain an injury of different tissue types, the more blood flow that you have, the faster the recovery time because it's bringing nutrients and getting rid of waste. So keep that in mind that if it doesn't have a lot of blood vessels, it takes longer for it to heal. Now, when we're looking at the functions of connective tissue, since there's so much variability to it, when we're talking about connecting organs to one another, tendons and ligaments are key pieces of those connections between bones and muscles. And then as far as support goes, bones and cartilage have a supportive function for our bodies. As far as physical protection, the actual cranium and ribs and sternum are all protecting essential organs such as the brain and your lungs and your heart. There's the immune protections, which are coming from your blood cells, specifically your white blood cells. Movement comes down to bones, and we're going to focus more on bones in chapters 7 and 9. And then storage, there's fat that also stores the calcium and phosphorus within your bones. There's heat production, which is dealing with metabolism of brown fat. And then there's transport, which your blood plays a role in. So if I'm looking at each of the variations that are pictured here, you will notice that you can see cells that are more spread apart. So in the adipose tissue here, you can see the smaller purple nuclei between the fat droplets. The fat droplets act as a storage component, but it's also an energy storage. It's functioning in protection as well because there's this cushioning that is involved with it. The extracellular material for your adipose tissue is literally the fat. With cartilage, the cartilage has this matrix that it produces surrounding the chondrocytes. Bone even though bone we think of being a more solid material, the bone cells, which are called osteocytes, are the little black dots you kind of see all throughout this image. And the extracellular material is actually a harder matrix made up of the hydroxyapatite. And then blood, people often don't think of blood as connective tissue, and it's because blood's extracellular material is actually the plasma, so it's a liquid, but the cells are more spread out. You have extracellular material. And then the fibrous versus the loose connective tissue, both of those have similarities in the fact there's collagen fibers, but it's based on the orientation of them. The fibrous connective tissue tends to be laid out more densely, and it produces your tendons and your ligaments, where the loose connective tissue, you'll actually see an example of this in the dermal layer of our skin. So connective tissue, cells are spread out. There's more extracellular material than cells, and they have a wide variety of functions. The other thing that I want to point out with this is I created this flow chart to show how we classify connective tissues. So connective tissue proper is dealing with the fibrous connective tissues, and there's loose connective tissues that include areolar, adipose, and reticular. There's dense, that's including the regular and the irregular. And then you have your class of supporting connective tissues. The supporting connective tissues involve your cartilage and your bone. The cartilage, there's 3 different variations that I'll talk about in just a moment. There's hyaline, there's fibrocartilage, and there's elastic. It's based on the extracellular material, how we classify those. For the bone, it does have a solid matrix to it, and we have 2 variations of bone. There's compact bone, which is literally more compact. The cells are tighter together, where spongy bone, it's more spaced out, and there's more sinuses that allow for distribution of not air, but more bone marrow in those areas. And then the third category is the fluid connective tissue, and that not only includes our blood because of the plasma, but it includes our lymphatic system as well, specifically the lymph, where you have a lot of white blood cells that move, but the lymph is actually a fluid that surrounds it. So as far as the different types of connective tissues that I really want you to focus your energy on, I want you to be aware of adipose tissue and be able to recognize it. Adipose tissue is essentially our fat, and you've got your adipocytes that are the dominant cell type, but the space between the adipocytes is occupied by not only fibrous tissue in the terms of areolar, reticular, but it also has blood capillaries. Keep in mind that this particular type of connective tissue is where our fat is stored, and it's the primary energy reservoir for our body. And the fat is continually recycled within our body. We synthesize new triglycerides and we hydrolysize old ones. So that way we keep that long-term energy going. There are actually 2 types of fats. There's white fat, which is the fat that's found in adults. It provides thermal insulation, and it has a cushioning function as well, not only for just underneath our skin, but our eyes and our kidneys. And adipose tissue also contributes to body contours, more specifically in females as far as breasts and hips are concerned. Now, brown fat is only found in fetuses, infants, and children, and its color is coming from the fact that the blood vessels are highly concentrated in these areas, and normally brown fat has a heat generating ability to it that helps with developing individuals. So adipose tissue, lots of functions to it. The extracellular material is fat. The adipocytes are the cells that are secreting the adipose or the fat, and it's used for storage, heat, and yeah. Okay, so let's switch gears to cartilage. Cartilage is a stiff connective tissue that has a flexible matrix to it. And cartilage gives shapes to our ears, the tip of our nose. It actually helps to support our larynx, which is involved in our respiratory system. And there are different types of cartilage cells, but we typically refer to chondrocytes as the mature variations of them. And when we're looking at the different types of cartilage, the way to distinguish them has to do with the type of fibers that are in the extracellular material. So elastic cartilage, like its name is giving away, it has an abundance of elastic fibers around it. And this provides flexibility and elastic support. We find this in our external ear and our epiglottis, which the epiglottis is essentially a flap that when we swallow, it blocks our trachea from getting food that's supposed to be going down the esophagus. Now, hyaline cartilage, which is pictured in the upper right-hand side, it has this clear glassy appearance to it. And it's because of the fineness of the collagen fibers that you find in the extracellular material. And where it functions is typically in articular cartilage, which is at the ends of long bones. You also find it in your costal cartilage, which is the cartilage that connects your ribs to your sternum. It's found in the trachea, the larynx, and in the fetal skeleton. Its functions are that it helps to ease your joint movements. It also holds your airways open when there's a pressure difference from the inside to the outside of your lungs. It moves your vocal cords, and it's also very important in growth of juvenile long bones. We'll talk about the endochondrial ossification when we get into the bone tissue in a little bit. So overall, the hyaline cartilage, again, is helping hold structures open and help with movement. Fibrocartilage tends to have coarse bundles of collagen fiber, so you see that it doesn't look as glossy as the hyaline cartilage. Its primary function is resisting compression and absorbing shock. This is found in the pubic symphysis, which is the joint between the left and right pubic bones, the menisci, which are the structures in your knee joints, or meniscus is probably the more common term you've heard. And they're also found in your intervertebral discs. So definitely areas for compression resistance and shock absorbance. The other thing I want to point out while I've got this picture up is that the chondrocytes basically get trapped in these little houses that are referred to as lacunae. And you see that in all three types of cartilage. And the reason I'm pointing this out is because those lacunae will start to fuse together. And when it's involving ossification or our body producing bone, sometimes it starts out as cartilage before it becomes bone. So speaking of bone, bone is a solid connective tissue based on the calcification process. And there's two major types. There's spongy bone, which it's called spongy because it has a porous appearance to it. And the compact bone, which is pictured here, it's denser and you can see the calcified tissue with no spaces between them. The compact bone is arranged in cylinders that surround a central canal, sometimes referred to as an osteonic canal, but this runs longitudinally through the shaft of the long bone, and there's blood vessels and nerves that travel through there. So that way the blood vessels can deliver nutrients to the bone material and bone cells, as well as take waste away. The other thing, the osteocytes are developed into these concentric rings because once an osteocyte produces the matrix around it, it basically traps that osteocyte into a little area. And the osteocytes will then have these little canaliculae, which are the little lines you're seeing. These actually are little tubes almost that connect cells to one another, so that way they communicate, nutrients can get to them and get out. And then the other thing that you'll notice with these is that each of the central canals looks like it's got rings around it, sort of like a tree. Each one of these rings indicates a new deposition of osteocytes that then get trapped. The outer layer of the compact bone is referred to as the periosteum. It's actually a tough fibrous connective tissue that covers the whole bone that helps to produce osteoclasts, which we'll talk about more in the bone specific chapter. So bone is considered connective tissue because of that extracellular matrix that is based on the mineralization of the material around your osteocytes. Now, the last specific type of connective tissue you're responsible for is blood. Blood is, again, a liquid connective tissue, and its ground substance is the plasma. Within the plasma, there's cells that move throughout. There's erythrocytes, which are your red blood cells. There's leukocytes, which are your white blood cells. There's platelets, which are involved in clotting. And there's other structures within the plasma too that include albumin and other material that needs to get transported throughout our bodies, such as nutrients that we're trying to absorb and use elsewhere. Hormones can also get transported in the blood plasma. So blood, the reason it's connective tissue is because all of the extracellular material is that blood plasma that has a lot of different transport functions. Now, the key difference between the types of formed elements within the blood, the red blood cells are responsible for transporting oxygen from the lungs to the rest of the body. It also helps to get rid of carbon dioxide. The white blood cells are responsible in our immune response. And again, those platelets are involved in our blood clotting processes. The next type of tissue that I'm going to focus on is what we refer to as excitable tissues, and it includes both the nervous and muscular tissues. With nervous tissue is including two major types of cells. There are neurons, which the neurons themselves is the cell that receives signals and transmits that signal to another cell, and basically will elicit some type of response to a stimuli. These are excitable cells because once the stimuli is received, there is this action potential that gets propagated through the cell to communicate to other areas of the body. Now, a neuron has some major components to it. The neurosoma is the main cell body of the cell that includes the nucleus. And this particular region is where most of the organelles are found, with the exception of some endoplasmic reticulum and mitochondria further down, and ribosomes. But the signals get received by all of these branches that are coming off of the neurosoma, referred to as dendrites. The dendrites receive the stimulus, and then they propagate that signal down a single essential extension that goes to the rest or to the next cell that it's trying to communicate with. And that extension that sends the signal to the next cell is the axon. When we get into nervous tissue in module 4, actually it's, I believe module 6 is our first encounter with it, but it's a little bit later this semester that we'll focus more on the overall structure of your neurons, because there's lots of different types of neurons, but they all have dendrites, they have neurosoma with the nuclei, and then they have the axon that gets the signal sent out. Nervous tissue is also made up of what we call glial cells or neural glial cells. Neural glial cells are the cells that can actually move around, and they offer all these protective functions with the neurons, because once a neuron is formed, a neuron is stuck where it is, and It has the primary job of just sending signals from one location to another. Technically, nervous tissue is made up of about 90% glial cells and 10% neurons. So the neurons, although they have a huge function in communication, the glial cells are what allow them to continue functioning because they're constantly being activated. The other type of excitable tissue, instead of just sending signals from a stimulus to where that signal needs to go, is muscular tissue. And muscular tissue is excitable in the fact that it can contract. So the cells that have this movement to them are excitable. Now, when a muscle cell gets excited, it will contract. And when it's able to have the stimuli no longer being received, it can relax, and then the process will repeat. Now, there's three major types of muscular tissue. There's skeletal muscle, cardiac muscle, and smooth muscle. They all have unique characteristics, but there's also some shared characteristics between them. Cardiac muscle and smooth muscle both are involuntary, meaning that we do not have conscious control over our cardiac muscles contracting, which would be our heart, nor do we have conscious control over our smooth muscle, which is basically the muscle that is lining any organ involved in our digestive system. So cardiac muscle and smooth muscle, we do not have conscious control over them. Skeletal muscle, we do have conscious control over, and the skeletal muscle is actually more organized. And when I say organized, I mean how the cells are oriented in comparison to one another than the other two. Now, the similarity that you'll see between skeletal muscle and cardiac muscle is that they both have what we refer to as striations, which is this banding pattern that exists because of overlapping microfilaments, specifically actin and myosin. And actin and myosin, based on the way the striations are formed, allow these cells to contract simultaneously and in a very unified fashion. Now, skeletal muscle, in comparison to cardiac muscle, are these really elongated cylinders that when they go through mitosis, they do not go through cytokinesis. So you get a really long cell that is multinucleated, meaning there's multiple nuclei that each nuclei is controlling a separate section of what's happening in the skeletal muscle. And when you look at the cardiac muscle, each cardiac muscle fiber has its own nucleus to it. The other variation you see between these two is that the skeletal muscle, again, are these long tubes where cardiac muscle is more condensed and it branches, and the separate branches of the muscle fibers will connect to one another through these structures called intercalated discs. These intercalated discs are tight junctions from one cardiac cell to another that actually allow the movement of ions from one cell to another. So when the signal is received for a contraction, that signal can get propagated to the next cell it's connected to. And because of the multiple connections due to the branching of the cardiac muscle, is why our heart is able to contract very simultaneously and in a very set fashion, where the atria contract before the ventricles contract. The other thing that you'll notice is that the smooth muscle, in comparison to skeletal and cardiac, is lacking striations to it, and they look more spindly, where there is a central nucleus and then it comes to a point on either side with the plasma membrane. And that smooth muscle, instead of contracting in very distinct units, it contracts more like the ringing of a towel almost, where when you, in order to get smooth muscle to contract, it actually tends to be stretched first, and then the contraction is in response to the stretching, and it aids in this process called peristalsis that happens through our esophagus and through our stomach and small and large intestine as well. So muscle tissue, the reason why we consider it excitable is because when it receives a stimulus, it will then cause the muscle to contract. And there are definitely some nuances or differences between the different types of tissues. Now, the last section of this material focuses on tissue growth, development, repair, and degeneration. Now, there's some terminology that you are supposed to be responsible for, and your book didn't have any very distinct pictures with it, so I did find these images online, and I was hoping that they would help to make the terms make a little bit more sense. So this image right in the middle here is of 4 normal cells, and when we use the terminology for tissue growth, it can deal with increasing the number of cells that you have or increasing the size of the existing cells. The image on the left-hand side here that's referred to as hyperplasia is the process of growth through cell multiplication. So we went from 4 to 16 of them. Hypertrophy is dealing with the enlargement of pre-existing cells. So you still have 4 cells here, but all of them are larger. Typically, hypertrophy is what happens when your muscles grow through exercise. This is also what happens when you get an accumulation of body fat. An interesting tidbit that I forgot to talk about when I was talking about the adipose tissue is that people often get into the idea of liposuction. And liposuction, it actually does remove some of your existing fat cells, but if you don't change lifestyle habits, what ends up happening is the fat cells that are left behind will just go through hypertrophy to fill up the space that is now available to them. So we have this pre-existing amount of fat cells that we will ever have as an adult typically, and what causes them to get bigger is more accumulation of fat per cell. The other things that you'll see up here is that down at the bottom image here, this is referencing how this sort of relates to cancer, where if you have a cell that develops a mutation and that mutation causes hyperplasia, which is the uncontrolled cell division, you'll start to get into this mass that is created, which can develop the tumor or the neoplasia. And then the dysplasia is when you start to get this differentiation in cell size and type in that given area. And then the in situ cancer is essentially where the tumor is still isolated. It hasn't started to become invasive yet. And when you have it grow to the point where it comes in contact with a blood vessel or a lymphatic vessel is when you'll start to get the metastasizing happening. Now, atrophy is the one that's pictured in this upper left-hand corner. Atrophy is what happens to our cells. Often when we're not using them anymore, the cells will get smaller, and if they become too small, they become non-functional, and you are unable to reverse the atrophy if that begins. Now, there are some things that it could be based on being bedridden for a little while, and then when you start to use those muscles and cells again, you can start to develop them, but often atrophy, if it cannot get reversed, will cause the inability to use different areas of our body. All right. The other thing in this section that I wanted to address was stem cells. So stem cells basically are cells that become a precursor cell for different types of differentiation within our body. And the very basic understanding of this is that a stem cell will most often go through mitosis to leave behind a stem cell that can then continue to make more stem cells. And then one of the cells will become a precursor cell and develop into a different cell type and therefore tissue type. Now, this typically happens based on the location of the body and differential gene expression. That is what dictates what the cell will become. Now, embryonic stem cells versus adult stem cells have some variations to them in the fact that embryonic stem cells, they are totipotent, which means that they have the potential to develop into any type of fully differentiated cell. And these cells are of very early embryos. So before we start to have the germ layers developing, that's when they would still be totipotent. Pluripotent cells, they can develop into any type of cell in the embryo, but they cannot be an accessory organ of pregnancy. And they typically are forming from the inner mass of embryo cells, which would be the blastocyst component. So once the embryo starts to differentiate a little bit more, the cells have a little bit more targeted end product. With adult stem cells are cells that are undifferentiated that you can find in mature organs. Typically, you can find these in our blood marrow, our bone marrow, where blood is developing. And some of them are multipotent, that they can develop into 2 or more cell lines. Some are unipotent, though, that they can only turn into one type of cell. And some of the issues with trying to use adult stem cells as stem cells for replacing other tissues is that they've already been developed in a particular region of the body, so they have less potential of the different things that they can become. Now, the other part of this particular section was tissue repair, where damaged tissues actually can get repaired in 2 different fashions. So when I talk about damaged tissue, I'm thinking of like having a cut or a wound. What generally happens is you can have regeneration, where you replace the dead or damaged cells by the same type of cell, and this should restore normal functions. Fibrosis is when you have the replacement of damaged cells with scar tissue. The scar will hold organs together, but it doesn't necessarily restore function. You can see scarring of lungs and tuberculosis. You can also see scarring in the case of severe cuts and burns. When you're having the healing of a cut in the skin, the severed vessel will bleed into the actual cut, and it will bring in what we call mast cells and the damaged cells. And in that location, they release a histamine. The histamine will dilate the blood vessels, and it makes the capillaries more permeable. So that way, blood plasma can seep into the wound, and it would carry antibodies and clotting proteins with it. And then the blood clot would form, and the blood clot is literally essentially connecting the edges of the cut together. And the goal of the blood clot is truly to stop the bleeding and inhibit pathogens from spreading. And when we get a scab that forms on the surface, it's temporarily sealing that wound and blocking an infection. And then macrophages, which are white blood cells, will phagocytize or eat the tissue debris from the damaged tissue. And then you'll get new capillaries that'll sprout nearby that vessel. And those deeper portions of the clot get infiltrated by both capillaries and fibroblasts, and it will take the soft mass and essentially remove the blood clot. You'll get new collagen being produced, and after about 3 to 4 days of injury, this will start, and it'll last till about 2 weeks. And then the epithelial tissue in the surface around that wound would multiply, migrate below the scab as the tissue's regenerating. And then the underlying connective tissue, it can undergo fibrosis, where you'll get some scar tissue, and it may or may not show through the surface layer, which this is showing the epidermis versus the dermis. So you can see the scar tissue down towards the bottom of it. The injury may take anywhere from a few weeks to completely heal, or it could potentially last up to 2 years, depending on the severity of the tissue repair itself. So our bodies are pretty fascinating, but it's all about recruiting new cells into the area and protecting us from infections. Now, as far as what happens to tissues after they start to degenerate and the cell death side of things, we've already addressed atrophy, which is when your tissues are shrinking in size. It's normally because of lack of use, or it can be a normal process of aging. Chapter 29 does a really good job focusing on all of the aging processes of each of the organ systems. Now, the difference between necrosis and apoptosis. Necrosis is a pathological tissue disease, or I'm sorry, tissue death due to trauma or toxins or infections. There's 2 major types of necrosis: infarction. Some people may be familiar with myocardial infarctions, which essentially it's the sudden death of tissue when blood supply gets cut off. The other end of the spectrum is gangrene, which gangrene is when you have tissue necrosis due to insufficient blood supply. This is typically caused by an infection, and there's different variations of gangrene. If you know anybody that has diabetes, often when they go for diabetic checks, they're looking for wounds that would exhibit dry gangrene. It's one of the common complications of diabetes. When you're dealing with other variations of gangrene, you can have wet, which it's your internal organs will essentially start to liquefy because of infection. And then there's gas gangrene, which that happens when you get an infection from soil bacterium that starts to produce hydrogen bubbles within your tissue. So all of those are different variations of causing insufficient blood supply, and it causes necrosis of the tissues. Apoptosis is what we refer to as programmed cell death or cell suicide. It's a way that normal cells, once they've completed their function, they can be of best service to our bodies by dying and getting out of the way. And normally when a cell starts to go through cell suicide, it will create these little branches that break off, and you get these little pockets that then can easily get phagocytized by macrophages and other cells. Billions of cells in our body die by apoptosis because once their use is over, it doesn't do us any good for an older cell to stick around when a new cell can replace that one. So that covers all of chapter 5, and I want to make sure that you still are focusing on the review guide and the reading guide, as well as taking the time to look through the other videos that have been posted for you, the discussion boards, and the thinking about worksheets that allow you to process material more deeply. As always, reach out to your instructor if you have any questions.